So yes, I am Alex. I'm a biometrician in David's group on level 8, and I'm going to talk to you about the bacterial population in Queensland. So to give a brief background, I think of it as a battlefield. So over 165,000 Australians develop hospital-acquired infections a year. The worst thing is that they are resistant to antimicrobials, so it sort of makes it more difficult in terms of treatment options in that sense. But those drug resistant infections add about $16.8 million in excess costs to the healthcare system. And the four species that are sort of responsible for this are your E. coli, your Staph aureus, which is your MRSA, Enterococcus faecium, and your Klebsiella. What has been identified is that whole genome sequencing is a powerful tool to track outbreaks of these resistant bacteria, but also to guide infection control in designing their prevention measures. So the operation is that we have Queensland Genomics, which is a 5-year program funded by the state government. And the goal of it is to deliver the genomics of medicine into the healthcare system and translating that genomic data into meaningful information for clinical care. So how are they going to do this? And what they're going to do this by utilizing the collective expertise of the healthcare industry, research, and academic organizations. So if you go onto the website, you'll see a handsome man telling you that pathogen genomics is overhauling a way to prevent disease outbreaks. So they've sort of come up with this whole system, and I'm going to run you through it. So you have your resistant isolates getting screened and sampled and sent to Pathology Queensland. From Pathology Queensland, through their tests, they're able to identify the resistant bacteria. And these don't really matter, but they have ways to do that. And once they've identified resistant bacteria, they send it over to FSS for sequencing. And once it's sequenced, you have the genomic data coming into UQCCR as well as the Beaton lab. In parallel, you have Pathology Queensland providing the sample data of the patient and, for example, the hospital or where the site of where the sample came from into the system as well. And that is stored into REDCap. In another third part there, another category is the clinical data. This is the infection control data that's coming into the system as well. And this is very important, and I'll explain this later on. So you have these three sort of categories coming together and being, I guess, compiled into a genomic report to inform infection control for our 3 major hospitals here in Brisbane. So that's the huge operation that's happening at the moment, and it's been happening for a couple of years now. And in my head, these are the battlefield leaders. So if you know them, go and talk to them about it, because they've been doing it for a while and they put a lot of effort into this. So one question you probably have in your head is that, where do I fit in all of this system? And I think I fit in over here. So the genomic data is about 2,000 samples or a bit more. And sort of what I came to do, I'm sort of like a paratrooper in this, jumping in at the end of last year. And the mission was to extract information, both understanding the transmission of these pathogens, but also how they transmit antimicrobial resistance genes. And so that I'll talk about later on. But in my head, I have a goal of 2 internal documents, one detailing bacterial evolution, the second detailing plasmid transmission. And both of these will form infection control, but also members within the group as well as collaborators. So these documents are designed to provide clarity for more design down the track. So the first document, Bacterial Evolution, will include addressing a challenge, and these are outbreaks. So from my understanding, infection control constantly surveys their hospital to identify an outbreak in their system. And they do this by inferring recent transmission between patients. From the genomic side, we sort of look at this as the relatability between bacterial samples, otherwise known as genetic distance. And so I look for what is known as a single nucleotide variation, so 2 SNPs. And so you sort of have this scenario, pretend that each of these bugs was sampled from an individual patient, and you have this distance between them. You can infer that these two, there's been recent transmission between these two patients rather than this third one here, just because of the sheer number. What the challenge is that happens is that is when there's dominant strains outside of the hospital coming in, and it's from the community. So there's a bit of you start to play with numbers down here where they're somewhat similar, but you need to understand the evolution aspect to navigate. Unfortunately, you can't really separate outbreak strains and community strains clearly because of the noise, and it'll lead to misinterpretation. So you have to understand the evolution to get to a sort of understandable threshold to address this challenge. So that's bacterial evolution. The second one I'll be looking at is the second challenge is plasma biology. So antimicrobial resistance is frequently associated with genes inserted into plasmids. So plasmids are circular pieces of DNA that are separate from the chromosome within the bacterial cell. What's interesting about this is that this DNA can spread from one species to another. What makes it more complicated is that there's many types of plasmids, but also within one plasmid type, there's many lineages. But if you start to think about how this piece of DNA is transferred from one species to another, you start to ideally look at a network, and then it starts to get a bit more complicated. But the challenge is that in pathology, this is what they'll see. So for example, you have an E. coli, and it's seen as resistant to, say, resistance A. Then you have a Klebsiella, and it's also resistant to resistance A. But literature would tell them that resistance A is associated with plasmids. So the question keeps popping up, is there a common plasmid transmitting throughout in the population? Or are there multiple? And the answer is we don't know yet. We do not know. So to address these challenges as well as many more, I sort of had to dig deep and sort of did an internal review of what I know. And what I figured out is that I'll have to draw upon the basics. So this is the career development slide, and it's very handy if you do one of these to know where you're going. So first in my PhD, I did a bit of the basics of plasma biology, looking in that transfer of antimicrobial resistance genes. And the second one, so that's my second position, is postdoc, and that's looking at bacterial evolution and knowing the basics of looking at the phylogenetic tree, which is like a family tree for species. So you're looking at family tree and understanding transmission, and then formalizing that transmission using clustering. So a way to formalize groups. And I did that with Salmonella. My most recent position, and that looked into developing a pipeline and the basics of that. So a pipeline is a series of programs lined up with each other, and there could be like 20 programs, but in an efficient way to get your answer using the genomic data. So I'm using the basics of these to then move on to a project design. So to do this, I assemble a project design, and that's looking at multiple fields and combining them into one. So you have plasma biology, which is looking at that AMR transfer of genes. You have phylogenetics, looking at the family tree, and this gives you context for, I guess, the evolution, as well as transmission and the sharing of genes as well. Sample data is very important because you need more data than the actual genomic data. You need patients where they were sampled, for example. Characterization, gene presence, absence, and this helps me personally with the transfer of genes. Infection control is very important here because it provides context for transmission for one, why there are so many samples within one hospital, as well as when you look at genomic data, you need to understand it's not uniform. It never is, and I keep thinking it's uniform, but when it's skewed towards one hospital, for example, or one area, infection control practices will start to make sense because of that skew. Coding, so I use Python and R, but in my head, it's very essential to have these fields here because I'm handling a lot of large amounts of data to analyze. And if you don't have a sort of background under each program and how they link together, you can get sort of overwhelmed. And the last one here is effective and accurate documentation. So in my head, I need to write all of this down in a very sensible manner because I want to guide or help infection control, and you need some of this documentation to be effective for translation of information, as well as if I get this done, it'll help current and future investigations with members of the Patterson group to guide their projects, for example, as well as collaborations. Now, what is the best thing about this is that all of these fields have a specialist within the Patterson group or in collaborations. So in my head, you sort of think about it as like the Fellowship of the Ring, the Planeteers, because of like, powers combined, or the Avengers, but you get the point. So back to bacterial evolution, and this is basically going into the approach itself. The challenge is to define those thresholds to separate outbreak strains and community strains. What this threshold has to sort of collapse two parts of information is the species. So each species evolves at a different rate, and we have to sort of acknowledge that, as well as the epidemiological data on top. And this is, again, designed to reduce the noise from those outbreak strains for the community, just a bit of understanding and clarity. So here's a phylogenetic tree where evolution is going from the left here to the right. This is a case where there was an outbreak in Cambridge of MRSA in the neonatal ward. And so this just looks like a bunch of lines, but you have to provide a context with layering the information on top. So first we'll layer the genetic distance. So here we see two samples, for example, and they have 0 distance between each other. You see these two samples here, they're about 2 SNPs from each other if you go back and go up. That's well and good, but you need some sample data to put on top of it. And so we overlaid the data on top. And so you could see here, in the sample ID, you have the patients here, but you also see this huge group here that is associated with healthcare workers. Now, that you can visually see that there's a group happening here and it's sort of a cluster, but you need a way to formalize it. And so I sort of designed a program to formalize this sort of thing, this group along this phylogenetic tree. And this is what it looks like. It just helps you visually see that this blue bar here represents this big cluster. Now, the interpretation of this is that you have your patients here, your babies have MRSA, but the transmission, because of the evolution from left to right, you can somewhat infer that it's been, you have a transfer from the babies to healthcare workers here. And so using that, the combination of evolution, your patient data, and the family tree to set everything in sort of context from each other, as well as the programs for clustering, you sort of get this bigger picture, or a clearer picture. So that's going to do that. The second stage is plasmid biology, and I sort of have to keep this open because we're addressing this common plasmid idea, circulating, providing resistance to multiple species, as well as multiple lineages. So it's important to know the bacterial population within the region first to give yourself some sort of reference before you go into the complexities of plasmids. So the second part is the gene presence absence, and this is more of a guide because of the many types that you can sort of, each type generally has a piece of DNA where you can reference it from. So you look for that piece of DNA. And once you have those two, you sort of combine them and start to stitch together the family trees on top of each other to see the carriage or the resistance they've shared. So there's 2 of that, but even before you sort of look into that, is initial step, and it's developing your efficient workflows. I'm I'm a big fan of efficiency, but especially when you're handling lots of data coming in and out, it's hard to get sort of grounded because you get lost in all the details if you don't have efficient work systems going on. So currently, I'm doing a bit of coding, testing, and optimizing. Project design is important in this sort of phase because you have to sort of design yourself that you're in the best position to analyze the data, as well as documenting everything. So very important. So if you want a visual, this is what I look like. in terms of I'm going to be running into lots of amounts of data after walking mountains of development. So, and this is probably the perspective of the Patterson group and thinking like, what is Alex doing? But it is all for these two documents, and it's to extract or understand the transmission of bacteria and the pathogens within Queensland and the resistance genes. So one detailing bacterial evolution and the second one detailing plasmid biology. And both of them will have a sort of a detail of an idea of the best approaches and workflows to handle these large amounts of data for analysis. But on the second end of each one, we'll have a comprehensive data characterization to guide infection control, to give them an idea of what they're looking at, but also future designs and current designs for projects within the group, as well as collaborators, but also for like, for example, for grants. So I'm sort of chasing the idea of like two PhD theses, but I'm sort of chasing the feeling of at the end, I'll be able to say it's all done and it's all there for people to design from. From that, I think I spoke a bit fast because I'm a bit ahead of time. This is the acknowledgements. They've done a lot of work to get this up and running over the last 2 years, I think. And there's many more people under that. So that's me, and I'll leave you with my final slide. And that's all.
Excellent. Thank you very much, Alex. That was quite fascinating. It would be weird, wouldn't it, to sort of throw into the mix, you know, could you do the same for COVID-19, you know, SARS-CoV-2? Obviously, just a sort of a random question, you know, could you look at genes from viruses and do a similar sort of thing? Obviously, they don't have plasmids. There's lots of differences. But what are your thoughts there?
I mean, yeah and no. I mean, the way of bacterial genomics is a bit different to viral. I mean, they do draw phylogenetic trees, that family tree. And basically the clustering algorithm that I designed was from viruses as well. So you sort of can adapt it. You just need a few specialists from the viral side that have done genomics of it to sort of push it through. But it's possible, definitely possible.
Okay.
I have chat somewhere in my show.
So, Tom, do you want to just introduce yourself, because a lot of people wouldn't know you, and you know what your role is in this program?
Sure. Hi everyone, my name is Tom. I work with Brian currently within the BT.
Oh, Tom, you're cutting in and out a little bit.
I forgot my microphone.
Ah, good thinking. Okay, let's have another crack.
Sorry, again, my name is Tom. I work with Brian. We are currently within the Beatson lab and hope to be joining you all soon. I am a bit of a software engineer as well as being a bioinformatician, where with Brian's lead and like his development with the various bioinformatics, we have developed several systems exploring this, particularly looking at like we have the Kathy system up right now, which I believe if I go to it, there is a video for it, even if you may not have access to it. And that is a previous version. We're currently working on an even better, more interactive version.
Thank you, Tom. Questions for Alex? You might have stunned everyone into silence, Alex.
Probably. They're probably thinking about it and be like, I need to ask something. I have something brewing. I'm here all the time. So if you have any other questions later on, please do ask. I probably can find the answer for you.
David, it's Jason Roberts here.
Yeah, Jason, go for it.
Alex, I really liked a lot of the slides that you had there and the various pictures. Could you tell me which of those movie series is your favourite?
Well, see, I think it'll be, at the moment, it's Lord of the Rings, but I have a nostalgia for, Captain Planet and that phrase, powers combined. So I'm just going to go Lord of the Rings out of all of them.
I thought it was a very good way that you brought those in, and it's a great way to engage the audience, and I thought you did that very well. So congratulations.
Thank you. Yeah, it's sort of something I've been thinking about, to give a point of reference of combining those different fields together. It's very, very important.
Yeah.
All right, and Amanda just sent you a lot of chuckling in the background, so that's good.
That's good. That's good to hear.
All right, well, thanks everybody. Stay safe. It sounds like our environment is becoming safer and safer. I saw somewhere today that Australia was ranked #1 in the world for its response, so that's pretty awesome. Alex is waiting for rugby to return to the world stage.
Oh yeah, I am.
And we'll all reconvene in 2 weeks. Okie dokie. Thanks everybody. Good on you. Bye bye. Good on you, Alex.
Bye.